everyone. Today I'm going to go over a quick overview of how to make our complementary bases with replication, transcription, and translation. So in the first step in replication, we have DNA. In DNA, here is the strand. I have already unwound it from the double helix, and now it is in a straight plane for us. So our first step is going to take the helicase, which is an enzyme, and unzip the DNA strand so it can replicate and double. So remember our DNA strand, T-thymine will bond with adenine, adenine to thymine, adenine to thymine, thymine to adenine, guanine to cytosine, cytosine to guanine, cytosine to guanine, guanine to cytosine, adenine to thymine, thymine to adenine, and thymine to adenine, and adenine to thymine. So now I've got two strands of DNA, strand number 1 and strand number 2. Okay, so I'm going to work with strand number 2 today. So I'm going to take strand number 2 and put it over to transcribe into mRNA. So here I had T to A to T, G to C, C to G, A to T, and T to A. All right. Now remember, in mRNA, it needs to become smaller because thymine is too big to go through the nucleotide pore, the nucleopore. So what we need to do is transcribe that into mRNA and use uracil instead. So just like DNA, this is going to double in the amount. So we'll end up with 2 strands, but I'm only going to do this part of the strand just for time's sake. Okay, so we are going to go ahead and make our mRNA. So our helicase would come in and it would separate the strands and start working with one of the strands to make it into mRNA. So I'm going to go ahead and erase this part of the strand just for clarity's sake. All right. So adenine, it instead goes into uracil because uracil is smaller and can go outside the nuclear pore to the ribosome. Thymine to adenine, cytosine to guanine, guanine to cytosine, thymine to adenine, and again adenine to uracil. So now I have my mRNA strand. Okay, so our next step is to still get rid of that thymine. So I need to transcribe it and translate it into tRNA. So we will do that on the next slide. So here I have my mRNA strand that I brought over from the previous slide. And we are going to make our tRNA. All right. So again, we've got our enzyme that's going to separate that strand, and the tRNA is going to work from this strand on the right. Right here. tRNA is going to work from that one. So we're going to find our complementary base pairs for that tRNA. All right. So tRNA, whenever I match that up, we've got U to A to U, C to G, G to C, A to T, and T to, oops, not A to T, almost lost it. Remember, it goes A to U, and then T to A. So now I have my tRNA represented by my black ink. Okay, now the strand is ready to be translated into amino acid chain. Remember, we separate those up into codons, which are 3 base pairs to make 1 amino acid. And it looks like we've got 2 amino acids to translate. So the first one is AUG. So our first position is A, that means it's in this block. Second position is U, means it's in this block. Third position is G. So that means we come over and we've got MET. All right, next one, CUA. And we do that in the same manner. First letter C, then U, and A. And we are left with leucine. And that is how we do replication, transcription, and translation.